Thank you very much, Peter, for the introduction. First, I would like to thank ABSS. I'm actually truly honored to receive this award for my PhD thesis, and also thank my two amazing PhD supervisors for their great support, which still continues, actually. Today, I want to talk about two main projects of my PhD, both of which are focused on TMS EEG technique. The first one looks at the contribution of sensory inputs to TMS evoked potentials, or TEPs, and the second one examines the physiological meaning of TEPs. And finally, I will briefly touch on the project that I'm working on at the moment. We can look at EEG recordings in response to TMS from different perspectives. For example, we can look at the recordings of each channel in time domain, and we can see a sequence of deflections in the recorded potentials, and they're very reproducible. We can also look at the recordings from all electrodes at each point of time and look at the spatial distribution of the potentials, or also get into more complex measurements and look at the source of those responses at the level of cortex. We can also assess TMS EEG recordings in the frequency domain by time frequency decomposition of responses at each electrode, and also look at the distribution of power at each frequency or frequency band across scalp. Of course, all these measures are interrelated, and they are just looking at data from or signals from different angles. To date, numerous studies have looked at TMS EEG signals to examine different aspects of brain dynamics and cortical network properties and the relationship with, for example, normal or pathological behavior. We certainly led to very important discoveries in the field. But despite all the great potential, there are two major challenges facing TMS EEG technique, complicating the interpretation of findings. The two studies that I want to present today tackle some aspects of those two challenges. The first one is that the results are subject to the key assumption that the recordings actually represent the direct interaction of the cortical of the stimulated area and TMS. What does it mean? We know that raw TMS EEG recording is actually a noisy signal. So, but thanks to the recent advances in detecting and removing artifacts, we can suppress the major artifacts in TMS EEG recordings. And the assumption is that the recovered signal is now direct cortical responses to TMS. However, recent findings suggest that this might not be a valid assumption. TMS is accompanied by click sound and coil vibration, which can interact with our sensory system and induce auditory and somatosensory evoked potentials, which can distort direct brain responses to TMS. To solve this, the common practice is getting the participant to listen to a white noise and applying a layer of foam between the coil and the scalp. These recent findings suggest that these measures are not enough. For example, a very recent study by Angel Petrojev's group showed that despite adopting all the online sensory suppression methods, increasing TMS intensity, which means that increasing the click sound, enhances auditory responses in the bold signals, as you can see here. Also, an earlier response or earlier study in 2015 showed very high similarity between the responses to the stimulation of visual cortex and a control condition. So a control condition was stimulating over shoulder, which could kind of trigger auditory and somatosensory evoked potentials. Also, a more recent study by Hartwig-Zebner's group used a more realistic sham condition over the scalp, mimicking auditory and somatosensory sensations evoked by real TMS without stimulating cortex. And they also showed very high similarity in a spatiotemporal pattern of the two responses, both real and sham conditions, across post-stimulation time point, across the whole post-stimulation time point. So in this study, or the first project, I aim to characterize and minimize the contribution of those peripheral evoked potentials or TEPs or PEPs in my TEPs. We recorded 24 participants stimulated motor cortex as a real and shoulder as a control condition, just to induce a general sensation, as I said, evoked by TMS. And we recorded EEG using 62 channels. First, I looked at the temporal correlation of the two responses at 3 different time windows. And here, the channels highlighted in white shows that showed significant correlation between the two conditions. And as you can see, they were highly correlated in frontocentral areas, and the electrodes around the site of the stimulation. Here, you can see that they were actually independent or showed the least correlation. I also compared the two signals in space domain. First, I used a cluster-based permutation analysis to compare the signals at each point of time. And here, the highlighted electrodes showed the where and when actually real TAPs were stronger. And again, as you can see, except for frontocentral areas and very early responses, all the electrodes were showed in TAPs were actually almost stronger in all of the, always stronger than PEPs. I also measured their spatial correlation at each point of time, and again showed that we had high correlation between the topography of the two signals after 60 milliseconds. Just to further the significance of the issue of contamination of TEPs by PEPs, here I show you what we recently found in our new working memory project. We simulated three different sites where actually have been shown to play important roles in working memory, and tested how TEPs or how PEPs actually affect the site specificity of our recordings. As you can see, responses to TMS over three different sites were very similar to each other, and also to sensor responses. In a space domain, you can see that after 60 milliseconds, the responses are actually almost identical. Importantly, in agreement with the findings in the motor cortex, we showed that the pattern of early signals were actually very specific to the site of the stimulation. So this is DLPFC, frontal eye field, and posterior parietal cortex. And you can see they're very specific to the site of the stimulation. Now getting back to the study. As a next step, we wanted to design a filter to suppress the sensor signals in the recording. We employed three different filters, which showed high efficiency in suppressing other types of artifacts in TMS EEG recordings: linear regression, independent component analysis, and SSVC. So I'm sure you're familiar with the first two techniques. The third one is actually a spatial filtering method, originally designed to remove TMS-evoked muscle artifacts. But here, for the purpose of this study, instead of using the frequency properties of muscle artifacts to kind of estimate the projection matrix, I use PEP data to estimate the artifactual or introduce PEPs to the filter as an artifactual component to be removed from TEPs. So this is the original signal, and these are the output of my filters. And now the difficulty is comparing these methods, because we do not have a ground truth we didn't have ground truth to benchmark the performance of signal of these filters against. So we base our evaluation on our own findings, so that a good filter should clean the contaminated area from the central area, if you remember, after 60 milliseconds, while keeping the unaffected area or the site of the stimulation intact. I took three main approaches for the assessment. First, measure the correlation between responses pre and post each filter, and the channels highlighted in white show the significant correlation between a pre and post or no correction. As you can see, linear regression showed high correlation across all channels, which means that so there was almost no correction, and SSVC found to be the most aggressive one. But the good thing about SSVC was that the changes were according to the pattern of contamination. So it kept the site of the stimulation intact or unchanged, but clean from the central areas. I also looked at the signal at the source level and found that when the similarity is maximum, for example, at 180 milliseconds, only SSVC corrected the signal and actually shifted the activity away from the contaminated area or overlapping areas to the motor-related areas. I also evaluated the filters based on dipole fitting method, which is actually another method of source reconstruction. According to the strong evidence in the literature, we know that the earliest responses to TMS are localized to the site of the stimulation. So a good filter should keep the source of the stimulation close to the site of the stimulation, and also because the source is localized at the earliest time point, the dipole fitting method should give us a high goodness of fit. So so here you can see this is the site of the stimulation, and these are the dipole detected in linear regression, SSVC, and ICA. And you can see that ICA and SSV, and this is the original data. So you can see that SSVC and linear regression in SSVC and linear regression, I found the dipole very close to the site of the stimulation. But ICA actually didn't, the source in ICA was actually very far away from the site of the stimulation. So it was a poor performance of this filter. But how about the goodness of fit? This is how I compared goodness of fit across different data sets. The first row shows the recorded data, original or filtered, and the second one shows the projection of the signals over the potentials from the detected source. So the more similar these two are to each other, it means that the highest goodness of fit the source has, or the filter has. As you can see, the two topographies look very similar in the original data and SSP set. So they show that they had high goodness of fit. If you see here, both of them had high goodness of fit. And among all the three different filtering procedures, only SSPC actually significantly improved the goodness of fit compared to the original data. So putting all these pieces of evidence together, we concluded that SSPC provided good trade-off between preserving early components or early TEPs while correcting contaminated components. And I've made code and data for this study publicly available, and these are the publications from this study and a letter addressing this issue. So if you're interested to learn more about this issue, please have a look. I also implemented the automated function of SSPC into Tesla Toolbox. So if you're interested, you can use it to use SSPC to remove the muscle artifact in your TEPs, and also you can use it to suppress any kind of control data from your real simulation condition. Now I briefly talk about the other challenge, or another project which actually addressed the other challenge in interpretation of TMS EEG recordings, and share some of our findings with you. As I mentioned, the main purpose of combining TMS and EEG is studying the dynamics of different cortical regions. And one aspect of dynamic is interaction among neurons and interneurons within cortical regions. So many studies have been designed to uncover those interactions, for instance, by looking at pharmacophysiology of each peak. For example, this is a nice review of the literature by Ghazal Darmani from Olf Siemens group on this topic, showing the contribution of different neurotransmission to the generation of each peak. We also conducted a pharmaco-TMS EEG study in collaboration with the same group, investigating the effect of excitatory neurotransmission mediated by NMDA receptors, especially to earlier components in TEPs. Here, actually, we didn't really find strong evidence for NMDA receptors, but the point that I'm trying to make here is that this type of studies mainly target the underlying mechanisms, which are very important, but do not directly tell us whether the net effects are toward excitation or inhibition. And as we know that changes in excitation and inhibition is an important aspect of brain dynamics and can be highly relevant to the changes in behavior. So it's very important to understand whether each peak or each component in TEPs are showing changes in excitation or inhibition. However, those of you who are familiar with EEG know that polarity in EEG, actually EEG signals don't indicate whether the changes are towards excitation or inhibition. For instance, we can have excitatory input to pyramidal neurons, and depending on the location of synapses, we can see either negativity or positivity in our EEG signals. And it can happen, that is the same for inhibitory inputs. And because we do not know the exact configuration of synapses following TMS yet, we cannot easily interpret the deflections in EEG. And this is why we always need to benchmark EEG signals against other measures of cortical excitability like behavior and MAPs. In our second project, we compared TMS EEG responses with cortical measures from paired pulse TMS EMG. So why we compared these two techniques, or the outcome measures of these two techniques? Because theoretically, both techniques are designed to measure TMS evoked intracortical excitability across time with minimal contribution from spine. We know that single pulse TMS EEG in single pulse TMS EEG technique, we have a continuous recording of activity following TMS across time. And because the recording is from the scalp, it is least contribution from spine. And in paired pulse TMS EMG, actually, we apply 2 pulses with a certain interval and see the changes in time, but only in a single time point. By applying a paired TMS pulses with different ISIs, we can plot the changes in cortical excitability and see this pattern similar to what we see in EEG. So based on this, and because we know that EMG signals are directly related to excitation, we can use EMG to sort of translate EEG signals. So we do not have very continuous data here, but we can see a pattern here, and our hope was, or our hypothesis was, we can actually translate this by this, and they are actually showing the same pattern. Again, I'm not taking you through all the comparisons that we made for this study, but what we did in general was correlating the amplitude at each point of time, amplitude of the two signals, and also compare the pattern of changes over time using the shape similarity algorithm. In contrast to what we had hypothesized, the two measures were not correlated at any of the evaluated time points or in their shape across time. So obviously, the first interpretation was that the two methods are sensitive to different neural populations. EMG responses are actually sensitive to a small population of neurons in motor cortex, while EEG signals are almost from everywhere. However, we dug deeper into it to see if there is any confounding factor playing a role here. And we found that actually there is another source of sensor artifact here, contaminating both types of signals or both measures, but in two different ways. And this artifact was sensor input from muscle contraction. How we found this? In paired post-TMS EMG recording, we separated trials into low and high amplitude of motor evoked potentials induced by conditioning stimulus, and found that when we have larger muscle contractions following the conditioning stimulus at the beginning here, TMS EMG methods or paired post-TMS EMG with lower ISIs or at the earlier time points show higher cortical excitability or larger cortical excitability, probably due to the spinal excitation. But in single post-TMS EEG, the story was a bit different. In these recordings, when I separated the trials into high and low motor evoked potentials, the cortical responses got stronger after 45 milliseconds, apparently when sensor signals actually reached the cortex. And this is the result from cluster-based permutation analysis, and you can see that the changes mainly happen around the sensory motor areas, which were very important for us in this comparison. So we can conclude that these two signals are differentially contaminated by the same factor, by the same artifact, so they are diverged by this confound. So as the next step of this study, which is the last step that I'm actually presenting here today, we looked at the spectral features of TMS EEG recording. Actually, we wanted to link the two projects. We wanted to see the sensory artifacts and also how these sensory artifacts can affect the relationship between paired post-TMS EMG and single post-TMS EEG. But we focused on frequency domain data. So first, we decomposed TMS EEG signals from both real and control condition into the frequency component, and found that responses actually happen at two different frequency bands, below and above around 12 Hz with distinct pattern. And this is the result from motor cortex TEPs, and this is from control condition or multisensory condition. And this graph actually shows changes in the power of oscillation at the site of the stimulation, and these are changes in Hilbert power actually. So we compared these two patterns between the two signals and found that actually there is a or significant shape similarity between the low frequency changes in low frequency power between the two signals, but the high frequency power actually were significantly different in shape between the two signals or the two responses. Actually, high frequency TMS EEG signals showed further independence from control signals. For example, they were not correlated at the site of the stimulation. So this is a site of the stimulation, left motor cortex. There were no channels showing significant correlation with the control condition. They were also they showed high site specificity when stimulating different cortical regions, which was very important. So this is, you can see that we stimulated, for example, posterior parietal here, DLPFC here, FEF, and this is the control. So it was it was very high site specificity for this response. And also, they were not influenced by changes in the amplitude of muscle contractions as we had seen in the broadband signal before. Most interestingly, in motor cortex responses, we found that changes of power at this frequency band were actually highly correlated or similar in shape with changes in cortical excitability observed in paired post-TMS EMG. which was actually the original question of the second study. So this is actually, we found out, okay, so we can use this to sort of, again, to translate our TEPs. And apparently changes in high frequency TEPs are similar to changes in the cortical excitability that we see in paired post TMS EMG. So based on all these projects, the two projects, I can make this conclusion that TMS EEG signals actually contain both TEPs and PEPs. And PEPs, or peripheral evoked potential, cannot be prevented if we use standard experimental method. And if we use these experiments, the standard methods, we actually need realistic control conditions to be implemented in our TMS EEG experiments. And we showed that I showed that SSPCR actually holds promise for isolating TMS evoked neural activity from the sensory signals. And high frequency oscillations probably reflect changes in the excitation of the site of the stimulation. And the main conclusion from these two projects, and also from all our experience with TMS EEG, we can say that if we want to uncover brain dynamics following TMS in our EEG signal, we need really further methodological improvement and physiological investigations of outcome measures. And this is what we exactly are doing in our current project. We are actually, we tested a large cohort of more than 150 individuals and looking at TEPs from at different levels, from behavior to molecules and genetics. And we are trying to understand, hopefully develop a better understanding of TEPs, their inter-individual variability, their region-specific responses, and their electro and neurophysiology. And then I wanted to thank you for your attention, and I want to thank all the collaborators of this project, especially Dr. Najib Rogash, who was the main guide, who has been the main guide for all of my research. Thank you very much.
Thanks, Marna. That was a wonderful presentation and such an impressive body of work. It's no surprise that you're the award winner for the best PhD. That was just brilliant. So we've got 5 minutes or so for questions. Please get your questions in the Q&A box. I just maybe wanted to start out by asking what is hopefully a simple one. Presumably you get more or less interference or artifact from some of these PEPs, depending on the site of stimulation. Can you give us some recommendations in terms of where are some of the better places to stimulate on the scalp or over the cortex?
Yeah, of course. So actually it's a very good question, but so yeah, the thing is it really depends on what kind of artifact we are looking at. So for auditory or some of the sensory, especially auditory evoked potentials, it might not be very different. Maybe if we get closer to ears, it might be stronger. But for example, for some other types of artifact, like muscle artifact, it's good not to be around the periphery or around, for example, for DLPFC, it's more prominent. Muscle artifacts are more prominent than the midlines. It really depends on what artifact we're actually focusing on. For my study, I really didn't find, for example, if we look at just peripheral evoked potentials, I cannot say there's much difference across different sites. Or at least in the pattern of contamination.
Great. Thanks, Marna. Any questions for Marna in the Q&A? While we're waiting for them to come through, I just wonder whether you see a role for these sorts of outcome measures clinically. Do you think that we'll ever get to the point where they'll be sensitive enough to be able to detect changes in excitability as a result of a therapeutic application, and might they be prognostically useful?
So this is actually, this is a very good question. And this is actually our hope, to have a better understanding of peaks and TEPs, and hopefully if we understand whether they are showing excitation or inhibition, we can translate that into behavior and changes in behavior. Yeah, definitely this is our main purpose. Yeah.